Okay, so let me get on with the preamble to Mike, who's doing the heavy lifting for the presentation tonight. So we start with Matthew 24:37. As it was in the days of Noah, so it will be at the coming of the Son of Man. And so we're going to look at what Scripture says, and then Mike is going to hit us with some really good information of what the science world is doing. Genesis 6, 1 and 2, this is the beginning of the description of the flood. And it came to pass when men began to multiply on the face of the earth, and daughters were born unto them, that the sons of God saw the daughters of man, that they were fair, and they took them wives of all which they chose. So the sons of God took the daughters of man as their wives. And then in Genesis 6:4, it says giants, and sometimes it's translated as Nephilim, or fallen ones, were on earth in those days, and also afterwards, when the sons of God came to the daughters of men, and they bore children to them. Those were the mighty men who were of old, men of renown. So whatever the offspring were from this union between the sons of God and the daughters of men, they were mighty men. And then 6:5, the Lord saw how great the wickedness of the human race had become on earth, and that every inclination of the thoughts of human heart was only evil all the time. So God became angry. But Noah found grace in the eyes of God, of the Lord. These are the generations of Noah. Noah was just a man and perfect in his generations, and Noah walked with God. And I highlighted, Noah was just a man and perfect in his generation. We know from the rest of the Bible that no man is perfect. No man is perfect, no, not one. But it doesn't stop at perfect. Noah was just a man and perfect. It continues in his generations. So my interpretation of what that means was Noah was perfect with regard to his DNA structure. So that's how we pass on from generation to generation all of the traits that we have. Our eye color, the heights we will grow to, all of those things, the hair color, the color of our skin, a lot of that's inherited. So Noah was perfect in his generation. So this is showing that he most likely had uncorrupted DNA. And 7:23, every living thing on the face of the earth was wiped out, people and animals and creatures that move along the ground, and birds were wiped out from the earth. Only Noah was left, and those people and animals with him on the ark. So God told us that he deliberately killed all the animals. Why might they have needed to be killed? Because their DNA could have been corrupted. So I believe what this Genesis 6 is saying, there was corruption of the DNA by the sons of God. Some people believe that the sons of God are often called the angels, and sometimes the sons of God are called the generations after Seth, because he was the godly son of Adam and Eve. We don't know for 100% sure, but whoever that was, if they procreated and they made these men of renown, and there was evil everywhere, then the human genome had been infected. And so this was God's opportunity to cleanse that. And it could very well be, and I'll show you in a minute why I think that's true, that the animals also had altered DNA. So let me read this. I think this was very well put. We could never imagine that a time would come when mankind's DNA would once again be altered as with the giants or Nephilim in Genesis 6. The modern alteration would not be done through sexual contact with the sons of God, but rather through man's knowledge of the DNA code and the ability to alter it. The human DNA code was completely sequenced in 2003 when the entire 3.2 billion basis pairs were completely known. Immediately, manipulation, including splicing human DNA into other animals such as mice and pigs, began. Animals such as sheep were cloned. They had a cloned sheep called Dolly, as the first cloned sheep. But to this day, there has been no known case of humans being cloned. However, I read today, how prophetic, China just announced a cloned monkey. Chinese scientists cloned 2 monkeys. Humans may be next. So if the Chinese are tinkering with monkeys, they have no qualms about tinkering with humans. And I think personally, you know, when people say, oh, well, it couldn't have been angels or fallen angels, because we're told in the Bible that angels don't procreate. But it says when they say they don't procreate in heaven. It says they don't procreate in heaven. There's no need to, because they're eternal beings. But that doesn't say when they were fallen and they are demon and they demon possess a body, like we've read in many chapters when Jesus cast out demons from humans and they went into pigs, or from a possessed man that was tearing apart the chains they would bind him with. So there was demon possession. If those men who were demon possessed had relations with women, then what would the offspring be? So I just want to bring that up. Scientists tamper with man's DNA under the guise of increasing health by identifying genetic diseases and imperfections. In 2006, Great Britain opened what is called designer baby clinics. In these fertility clinics, 8 days after embryos are produced by the woman, they give them fertility drugs. And so they produce 10 or 15 or 20 embryos. And after the embryos are 8 days old, then they extract the embryos from the woman's womb, and then they examine them with different techniques to see if they might have some inherited disease. And then all of the other embryos are killed. Defective embryos are destroyed, and the best one is then reimplanted in the woman's womb. So if that isn't acting like God, selecting, first of all, you're killing 19 to get one. And the other thing, you're destroying human life, because life begins at conception. And then you're making the judgment of who lives and who dies. Natural progression is to enhance the human race by sharpening the senses. With the addition of eagle DNA, a man could see as an eagle. Or by placing deer DNA, the person could hear. Same could be accomplished for smell, and for strength, you could introduce gorilla DNA and give the person super strength. And how about some cheetah DNA to make you run faster? So all of this is happening, and the following quote I found is very telling to me. Humanity, this is a scientist who's working in the lab on this problem, their quote problem. Following quote is, humanity is on the verge of becoming a new and utterly unique species, which is called Homo evolvus. So let me turn it over to Mike.
All right, so in the days of Noah, I did this June 2016, the first set. So now we're going to do an update, which is going to cover some of the stuff you did. But the idea being here, in the days of Noah. There we go. So what did Jesus mean when he said, in the days of Noah? So it will be, so this is talking about when Jesus is talking to his disciples in Matthew. As in the days, so will be the coming of the Son of Man. So Bob and I think we are in those times. So what does that mean? So we went back as Bob did, and I won't go over this stuff. But what I will go into is now the earth was corrupt in God's sight, and the earth was filled with violence. And God saw the earth, and behold, it was corrupt. For all flesh was corrupt in their way on the earth. And God said to Noah, I have determined to make an end of all flesh. For the earth is filled with violence through them. Behold, I will destroy them with the earth. So, as Bob said, as Jesus said, in the days of Noah, there was corruption. And what is corruption? To cause someone to become dishonest, to change, less pure, or incorrect from the original form. So what we're talking about here is the beings on the earth were corrupt, inside and outside. As Bob and I will say, the DNA was corrupt. The only one that was not corrupt was Noah's lineage. And that's why God saved him with a flood. So where are we in the 21st century? As we've talked numerous times, the corruption of science is upon us. It's driven by money, not truth. It's amoral because the consequences are ignored because the world has become non-fearful of God, which is, and the Bible says, the beginning of all knowledge and wisdom is what? The fear of God. So throughout the world, socialism, communism, which is really anti-God. And we've had a reprieve in the last year in our country that we have the first time a president has actually gone to, some of the life movements. So he's taken us aside and he's bringing us back. Some people compare it to the Cyprus of the book of Daniel. So what does this mean? If we talk about the corruption of the species, corruption of the DNA, that means we have to go back to what is life. So back 2 years ago, I talked about some of the life indicators of fertilization. And I want to bring this back up, because if you don't remember, what was discovered in 2016 is when a zygote was fertilized by a sperm, there was a flash of light. The light of the world. Every time something became life, there was a flash of light. And I'm going to show you that right here. I don't know if you remember this, but the first visible sign that an egg is fertilized, this was newly discovered by a spark. So this is a couple minutes long. You can see it right now, right? So you'll see it. See that? The sperm entered the egg, flash of light, that's now live. And we, in this country, we've aborted, 70 million zygotes. So there's a big difference between dead and light. And this is what man is trying to become, as we are going to show with the Disney Sorcerer's Apprentice, is man now wants to step in and become a creator. See? All those flashes? That's life. That's the difference between dead and light. Dead and life is light. So with that said, one of the things is, how did life start? So there's a thing called DNA. DNA is a structure in your body. It was discovered in the 40s. Well, they found out the way to synthesize DNA is a thing called RNA. RNA builds DNA up from nothing. So the idea being that if you could find a mechanism that could create DNA from nothing, you could find how life was started. So I'm going to show you a slide from this professor. RNA is the origin of life. And I'm going to show you, it's a couple minutes long. Can we hear it?
This is one of the most important questions humanity has ever posed. And the scientific answer is, we don't entirely know. You might think that cracking DNA's genetic code should have explained life's origins. And it definitely helped. Thanks to our understanding of DNA, we can map out the history of evolution all the way back to single-celled life. But that's where we're stuck. The problem is, DNA is a great way to store information, but it doesn't do much else. Cells rely on other molecules like proteins to replicate, grow, and survive. Proteins, on the other hand, work great as molecular machines to keep cells alive and healthy, but they can't store information or copy themselves. They need DNA for that. So we have a chicken and egg problem. DNA needs proteins to function, and proteins need DNA to exist. So which came first? Which molecule made life possible? Well, there's a third type of molecule that may hold the answer: RNA. Most scientists think that RNA came first because RNA can do two jobs: store information and perform various functions that keep cells alive. This idea that RNA came first is called the RNA world hypothesis. RNA world suggests that billions of years ago, in some primordial soup of molecules, a self-replicating RNA formed. This may have happened in volcanic vents deep on the ocean floor, or perhaps clay clumps brought the necessary chemical building blocks together. Some scientists have even speculated that early RNAs formed on Mars and hitched a ride on an asteroid to our planet. One way or another, self-replicating RNAs emerged, multiplied, and evolved. Over millions of years, they developed into a legion of molecular machines. These microscopic proto-life forms blossomed and competed. The best collections of code lived on, and the weaker ones died out. Survival of the fittest was the name of the game. This competition for survival eventually led RNAs to evolve the ability to build strong, stable proteins, which excelled at carrying out complex biological processes. And somewhere along the line, some critical RNAs mutated into the familiar double helix of DNA. DNA became a stable archive of genetic information that stored blueprints for the most successful RNA and protein molecules. Life became more complex over trillions of tiny steps and happy accidents. And all the while, the RNA lineup grew, alongside lengthening genomes of DNA and complex proteins. And it's all still happening inside your body. RNAs have adapted to become the Swiss Army knives of our cells. Today, they can slice, dice, catalyze, build, destroy, code, replicate, and transform. A remarkable diversity from the simplest of beginnings, a single self-replicating RNA molecule.
Now, great in theory, right? If you notice, somehow it started. That's what he says.
Right, somehow.
So what I want to add to this, in the last 4 months, the guy that postulated this theory said, it's all wrong. All my postulation, everything's wrong. That is a huge acknowledgement that this is not true. The way in which he created the start of RNA has turned out to be false. So this is one of the first times in the last 50 years that they've admitted that it's wrong. So if we go back to the corruption of science and the chasing of money, one begins to wonder if that is because we have a president that's unfunding a lot of this ridiculousness from a standpoint of science. But the bottom line is that RNA theory has now been shown by the developer, the originator, as being false and wrong. So that's a win for what we believe, as we just saw. God created the mechanisms from nothing. And when there's two things become life, we see light and it's real. The other thing that we talked about last week, or last couple years ago, was marijuana. You know, it's now accepted in 27 states. This is a map. This map shows you the green, which is where it's for recreational use. If you notice, it's coast-based. And then you see the dark green, where it can be used for medical reasons. And then you have the rest of them where it's illegal. Now, as a country, it's illegal to have marijuana. So the states are usurping their power But the overall Fed is saying it's still wrong. That's why they can't put money in banks, because banks can't accept it. But the point of it is, in 2 years since I did this, it's become accepted by the citizens of this country. And there's many long-term effects within that pure DNA stream. So it's becoming corrupt, which gets back to the days of Noah. Now I want to give you an update on global warming. Now, the biggest fear of global warming is two things. One, all the ice melts because of all man-made heat. And by melting all the ice, the earth will flood because the level of the seas will raise 10 feet to 100 feet, which means where we live becomes unlivable. So that's the big fear. The other one is the oceans will heat up and burn. If we go by the first scare by Al Gore, 10 years ago, the oceans that we have out in the Pacific, instead of being very cold right now, would be boiling and steaming, and there'd be nothing left. So there is an issue with what's causing the ice to melt. Well, NASA just came out with this little winner. So let's watch this one.
Breaking. NASA drops global warming truth bomb. People are stunned. Progressive global warming activists will be disappointed at this recent revelation. A new NASA study has found that a geothermal heat source underneath the ice caps, consisting of heated rock and volcanic activity, is the real reason why the ice caps are melting, not global warming. Scientists have long speculated that a geothermal heat source, called a mantle plume, lies directly underneath a significant portion of Antarctica. This recent study, however, has explained how the ice sheet collapsed so rapidly in an earlier era of rapid climate change and why the ice mass is so unstable today. I thought it was crazy, said Helene Serapsy, the study's co-author and scientist at NASA's Jet Propulsion Laboratory. I don't see how we could have that amount of heat and still have ice on top of it, she said in an official statement. They found the level of geothermal energy from the mantle plume to be around 150 milliwatts per square meter. In comparison, under Yellowstone National Park, a geothermal hot spot produces about 200 milliwatts per square meter average throughout the entire park. Mantle plumes are thought to be narrow streams of hot rock rising through the Earth's mantle and mushrooming out underneath the crust. First theorized in the 1970s, the idea was offered to explain how geothermal activity could occur far away from the boundaries of tectonic plates. However, proof regarding the validity of the mantle plume theory has been accumulating for years. In 2014, researchers at the University of Texas found that Western Antarctica was a hotbed of geothermal activity, a conclusion confirmed by a team of U.S. scientists 1 year later. The high geothermal heat flux may help to explain why ice streams and subglacial lakes are so abundant and dynamic in this region, the study found. Even earlier this year, Scottish researchers found 91 previously unknown volcanoes underneath the Antarctic ice sheet, some over 13,000 feet tall, further corroborating this body of evidence. Scientists and global warming activists have been especially concerned regarding the melting of the ice caps. A recent discovery that a Delaware-sized chunk of ice split off from the ice sheet caused alarm throughout the scientific community. Chris Mooney from the Washington Post said that this change could draw further attention to the threat of climate change at a time when President Trump is considering whether to exit the Paris Agreement to reduce greenhouse gas emissions. Many other publications, such as The Guardian and Business Insider, published articles to a similar effect. Some scientists, such as Helen Amanda Fricker, don't concur with the mainstream appraisal of man-made climate change's impact on the ice caps. Glaciologists are not alarmed about most of these processes. They are examples of Antarctica simply doing what we know Antarctica has done for thousands of years, she wrote. Many might recall a video from famed meteorologist John Coleman, founder of the Weather Channel, where he spoke at length on the topic of how man-made global warming does not exist, and that climate change complex is a scam, although controversial at the time, isn't.
So, did you get that? What's causing the polar ice cap to melt?
Geothermal.
Volcanoes! Isn't that amazing? And were the volcanoes controlled by? The earth, by God.
Right.
So, the politicians are pushing an agenda, and in reality, if they put the thermometer in the water, they notice it's getting warmer. And it's not on top of the water, but it's underneath. So this is eye-opening, and this is proven. All the other stuff is controversial. Now, what I'm going to show here is a 26-minute, it was actually put together, I found it very interesting on YouTube. But this is the state of the world as man tries to become the creator. He's taking the mantle, he's taking the sorcerer's hat, and he's going to start running himself. So here are 8 things that are going on right now that happened in 2017 that are going to grossly impact Jesus' prediction of in the days of Noah.
Specified hazel eyes, dark hair, fair skin.
Designer children, engineered to perfection.
Would you drink human breast milk that came from a cow?
Human cells have successfully grown inside a pig embryo. Glow-in-the-dark cats.
De-extinction. It's not science fiction at all.
And scientists announced today they have made a major breakthrough in the quest to create life in a laboratory for the first time.
For the first time, scientists have created artificial DNA, the universal genetic code.
They have produced a living cell in which the active DNA was put together by computers. What does this mean for science, and does it mean there is now synthetic life?
Scientists at the Scripps Research Institute in San Diego, California, created an organism not found in nature. They did it by adding artificial genetic building blocks. DNA is the genetic material the body uses to store the unique blueprint for each human being. The information exists as a code based on 4 chemicals, nicknamed A, T, C, and G. They pair up with each other in different combinations to create 20 amino acids, the building blocks of proteins. The Scripps team created 2 artificial chemicals called X and Y. They inserted them inside the cells of simple bacteria. That created the potential for 172 amino acids and proteins that do not currently exist. Those proteins could be used to create a new generation of drugs and vaccines. Adriana Heggie is the director of the Genome Technology Center at NYU Langone Medical Center.
What I think is fantastic about this technology is that we basically were able to cheat evolution and to trick this organism into accepting molecules that do not exist in nature.
World-renowned geneticist Craig Venter has been trying to unlock the mystery of life for 15 years. What have you achieved?
We announced today the first synthetic cell. Instead of having a genetic relative that it evolved from, the parent of this cell is a computer.
What Venter has done is astonishing. With just 4 bottles of chemicals off the shelf in his lab, his team replicated more than 1 million bits of genetic code to create a living organism.
Here's how it worked. They isolated bacterial cells and removed all of their genetic material, the DNA from inside. Then they took those 4 bottles of chemicals and used them to create new genetic material. They transplanted it into the empty cell. That material, that new cell booted up and began to reproduce. And reproduce 1 billion times.
I think this is the creation of life, and I think it's an experiment that shows that life is not a mystery. That's a mysterious force that infuses things, that makes them come alive. You put the right genetic message in the right order, put it into the right environment, it will come alive.
But along with promise comes precaution.
Like any great scientific innovation, this has enormous promise and enormous peril. This may allow us to make more virulent viruses. This could unleash a bacterium on the world that has properties we didn't expect that could cause great disease or ecological damage.
Scientists will surely debate whether this is truly creating new life, but no one can deny the potential impact of this achievement.
Venture says that within a year, this technology will be producing vaccines, and in the future, bacteria may be used as powerhouses to generate energy and clean up oil spills.
The Salk Institute recently created a human-pig hybrid. Why? The research project studies the possibility of human organs grown inside a pig fetus for transplantation.
Yes, you heard that right. Human cells have successfully grown inside a pig embryo. This is one step closer to the possibility of growing livers and other human organs inside of animals.
Scientists have potentially discovered a way to stop genetic diseases from being spread to future generations. They did so by altering the DNA in gremlin cells, like sperm, embryos, and eggs. They plan to only use this new method to correct disabilities or diseases, and not to improve or enhance a human's ability or state of health. This finding is revolutionary for the future of healthcare around the world.
In labs across the country, they're working with a new tool that can precisely edit genes inside living cells. It's a genome editing tool called CRISPR-Cas9, and it's being described as a game changer.
I like to call it a molecular scalpel for cells. It allows scientists to make a very precise change in the DNA of a cell or organism that could do something like correct a mutation that would otherwise cause disease.
Jennifer Dowd, a molecular biologist at UC Berkeley, was one of the scientists who developed it.
It's a technology that just allows a level of precision that we haven't had in the past.
Now it's being used in laboratories across the country in plant and animal research. Scientists have their eye on using it for everything from coming up with cures for blood disorders like sickle cell disease to combating antibiotic-resistant superbugs. At this laboratory outside Washington, scientists are using it on zebrafish to knock out genes that cause deafness.
The technology is moving very fast. It's almost every scientist is interested in it. The concept of genome engineering has shifted to using these techniques because they work so well.
Harvard University scientists are even using the tool on mosquitoes with dreams of eliminating malaria. But the rapid embrace of the technology also has scientists concerned.
Just because we can build a CRISPR gene drive to spread the trait through the wild population does not mean that we should.
A top concern, editing so-called germline cells, meaning human sperm, eggs, and embryos, where DNA changes could then be passed down from one generation to the next.
Inside a converted shoe factory in Shenzhen, China, scientists have launched an ambitious search for the genes linked to human intelligence.
It must be over 60% of your IQ is decided by your parents.
60%?
Yeah, so we want to prove it.
The man in charge of the project is 21-year-old science savant Zhao Bowen.
We're looking for the smart people. Those that have an IQ higher than 145 points.
Zhao works for the Chinese biotech giant BGI, which owns more gene sequencing machines than anyone in the world.
It's just continuously generating tons of data.
Tons of data taken from the genetic makeup of more than 2,000 of the brightest people BGI could find, most of them scientists and math wizards.
And we want to see what's in common.
What part of their genomes are they the same?
Yeah, that's important.
One of the people selected and who volunteered his DNA is Boston software developer Jeffrey Kaufman.
this is something that, if executed well, could be very positive for our society and for the world.
But discovering the genetics of intelligence could also be controversial. It would then be possible to test a human embryo to learn if the baby is destined to be smart. In vitro laboratories already allow parents to use genetic tests to screen their embryos not just for disease, but to choose embryos based on gender and other physical traits. BGI believes that one day there might be a test for IQ. So in the future you're saying, yes, people may well select for intelligence.
The technology will be there.
If that day comes, Kaufman believes that children chosen for intelligence would have an advantage.
Given the choice, most parents are going to rather have a more intelligent baby than a less intelligent baby, sort of all other things considered, and that for the most part, that seems like it should be a good thing.
Yet the fear is that China could unleash a future of designer children, engineered to perfection, the stuff of science fiction.
You have specified hazel eyes, dark hair, and fair skin.
The 1997 film Gattaca envisioned the ethical dilemmas in a society that screened out human imperfection.
Taking the liberty of eradicating any potentially prejudicial conditions, premature baldness, myopia, alcoholism, and addictive susceptibility, propensity for violence.
Just this week, the U.S. government gave a conditional thumbs up to embryonic gene editing research. By moving or improving components of human DNA, this new gene editing technology could enhance someone's physical and biological characteristics, as well as eradicate a lot of diseases. Until recently, lawmakers had banned any form of gene editing.
In addition to the DNA of the mother and the father, material from a third person is used in the process.
The vast majority of the cell's DNA is located in the nucleus, but a tiny fraction is outside the nucleus in structures called mitochondria. These mitochondrial genes are inherited only from the mother and in rare cases can be defective. These defects can cause problems such as blindness, muscle disorders, and neurological illness. One proposed technique would work this way. A woman's nucleus is removed from her egg, leaving behind her unhealthy mitochondria. Her nucleus, containing the majority of her genes, replaces the nucleus in the donor egg, leaving an egg with the mother's nucleus and the donor's healthy mitochondria.
A baby comes into this world unlike any other baby has before. For the first time, doctors in Ukraine used a new type of three-parent IVF technique so that a previously infertile couple could have a child. To clarify, this isn't the first child born with DNA of three parents, but she was the first to be conceived using a technique called pronuclear transfer. In this instance, doctors in Kiev combined the father's sperm with the mother's egg and then placed those genes into an egg that was donated by another woman. Then they implanted that egg in the mother's womb, and on January 5th, she gave birth naturally to a baby girl. The baby has her parents' genetic identity with a trace amount of DNA from the donor woman. But her conception is controversial. For one, the science of three-parent IVF is new and still highly experimental. It also raises a number of ethical concerns and fears over the possibility of a future with so-called designer babies.
There are almost 7.5 billion people on Earth. They come in all shapes, sizes, and colors. 15,000 humans are born to proud parents every hour. But over the years, some have tried to find another way to create life. Human cloning.
You're copies of people out here in the world. Why clones?
Its possibility is proven controversial. 19 years ago, 19 European countries took it upon themselves to ban human cloning.
You can't just go around cloning people. That's crazy.
The practice is broadly seen as dangerous and unethical, and opponents say it could be prone to abuse, arguing it will lead to a generation of humans whose organs and tissues will be harvested.
The shroud of the dark side has fallen. Begun. The clone war has.
With the cloning of Dolly in 1996, the idea of human cloning became a hot topic of debate. To date, no known human clone has ever been produced, but in 2008, researchers cloned 5 human embryos. This was seen as the first step towards using cloning techniques for biomedical research and therapy.
book called The Birds. In it was a section showing the few U.S. species we'd already lost to extinction. The great hawk, the Carolina parakeet, the heath hen. The biggest picture, though, was reserved for the passenger pigeon. Once numbering 3 billion or more, this species flew in vast flocks, passing overhead for days at a time. Witnesses described seeing a feathered river in the sky. Our hungry, growing nation also saw them as delicious and inexhaustible, and hunted them to near extinction in about 50 years. In the book was a picture of the very last one, named Martha, stuffed and sitting on a perch in the Smithsonian Museum of Natural History. Next to her was a sign that said, extinct. From 3 billion passenger pigeons to none. I still can't believe it. Martha died 100 years ago on September 1st, 1914. Her demise proved to be a watershed event. People finally began to care about the fate of our nation's wildlife. Conservation groups sprang up, including those formed by hunters to protect habitat, allowing many species to flourish, including grassland birds and waterfowl. This pigeon story doesn't end there, though. In a scene somewhat out of Jurassic Park, scientists today are actually trying to resurrect this grand bird. Starting with DNA from preserved museum specimens, the passenger pigeon's genetic code will eventually be spliced into the living cells of a close cousin, the band-tailed pigeon, with the hope of creating something that looks, sounds, and perhaps even acts like the real thing. They're calling the process de-extinction, and it's not science fiction at all.
De-extinction is an effort to bring back species that are known to be extinct. Things like saber-toothed cats.
Ross McPhee is the curator of mammals at New York's American Museum of Natural History.
The screenplay for Jurassic Park 4, I'm sure it's already being written, because here we have science colliding in a way with our worst fears.
Scientists in Australia announced last week they've been able to recreate the embryo of a unique but now extinct frog. Scientists at Harvard are doing research that might allow them someday to bring back the passenger pigeon, a bird that populated the eastern U.S. in the early 1800s until it became extinct at the turn of the century.
Laboratories around the world are working to bring back other long-gone species as well, such as the Pyrenean ibex, the Tasmanian tiger, and the woolly mammoth.
The woolly mammoth has been extinct for about 4,000 years. It is now on the verge of being resurrected in a modified version. Harvard professor George Church claims his team within the next 2 years will be able to create a hybrid embryo with an elephant and mammoth DNA. The scientists are likely to be many years away from producing a living so-called mammothant, but a successful attempt of creating an embryo could just be around the corner. Church says this elephant-mammoth hybrid would not just have the mammoth's distinctive long shaggy hair, but also smaller ears and blood better suited for cold weather. Church believes these qualities could actually help save the endangered Asian elephant, but others worry it's unethical. The scientists hope to actually grow the embryo in an artificial womb, hoping to avoid risking the life of female elephant that is already endangered.
Would you drink human breast milk that came from a cow? In suburban Beijing, scientists have created a growing herd of over 300 cloned and genetically modified cows that can produce milk containing the same health properties as human breast milk. Dr. Li Ning, professor at China's Agricultural University, said that the genetically modified cow milk is 80% the same as human breast milk. Ning explains that the modified milk contains many of the same antibodies and proteins that are good for our health, can improve our immune systems, and can help us fight viruses and bacteria.
It looks like a horse, sounds like a horse, and walks like a horse.
Well, I've been around this since they started here, and they're amazing.
So much so that a retired professional rodeo cowboy will even vouch for them.
They're not creating monsters. These horses are as real and as natural as anything you've ever seen in healthy.
Clint Johnson owns a few Broncos that buck just like the famous horse he used to ride. Like these foals, they are clones, genetic copies made in a lab.
Hundreds of years ago, man decided to breed animals for an intended purpose, whether that was for recreation, for companionship, or for food. Cloning is just another step in that process.
Blake Russell is the CEO of Viagen. His Austin, Texas-based company clones livestock, including cattle, sheep, pigs, and horses.
Cloning is an amazing technology to turbocharge those breeding programs, but is it any more plain God than any of the other technologies?
The company collects tissue samples, preferably from a living animal. With the DNA from those cells, the lab produces embryo copies through a process called somatic cell transfer.
We named her Chloe, and she's 3 years old, and this is one of the reasons why we clone her. We take an animal that wasn't fertile, and obviously she couldn't produce any genetics. She's been to the national finals 19 times in a row since she was 3 years old, and here we have her back, and we can breed her to whatever we want.
In this case, competitive horses. Viagen's biggest share of customers are people that want to reproduce their polo, rodeo, or competition riding horses. The company claims its success rate in getting cloned embryos to newborn animals has climbed, but admits the process isn't perfect. Cloning competitive horses is a touchy subject. Only this year, the governing body over equestrian events okayed the use of clones in competitions like the Olympics.
South Korean scientists have created a glow-in-the-dark dog. Need I say more? Yeah, probably. So here's the story. The research team at Seoul National University made a beagle using somatic cell nuclear transfer technology. In other words, they cloned it. Tegan, as they named the fluorescent Fido, was genetically engineered to glow when placed under ultraviolet light.
People think that cloned cats are creepy, but what about a cloned cat that also glows in the dark?
It's an incredible story. Not everybody's happy to hear this, though. The scientists in South Korea cloned a pair of kitties who light up with a fluorescent glow and expose to ultraviolet light glow kitties.
All right, now the question is, what was all this for? Apparently there's a good reason for it. Christy Wustow has the story.
At first glance, these cats look like any normal cat, but there are two big things which make them very different. One, the cats are clones. Two, they glow in the dark. You heard right. When they're put underneath ultraviolet light, they glow a dull red. And it's no accident. Scientists manipulated the donor cat's genetic code, then passed those changes onto the clones.
It's meaningful as we introduce outside genes to transgenic clone cats for the first time, and as they have the red fluorescent protein gene in their organs, they give off a red color.
Here's how they did it. Scientists took skin cells from a Turkish Angora female cat and used a virus to insert the genetic instructions for making red fluorescent protein. Then they put the gene-altered nuclei into eggs for cloning. After that, they implanted the eggs back into the donor cat, which effectively became the surrogate mother. Now, these glow-in-the-dark cats shine a light on what may be possible down the road.
A company has tinkered with the genetic material of salmon so the fish can grow faster and reach groceries sooner. Are eaters ready? Business to start.
It looks like any other salmon, but opponents call it frankenfish and hope to keep it off your dinner plate. Here's the difference. Both these fish are 18 months old, but the larger genetically engineered Aqua Advantage salmon grows twice as fast as the regular salmon.
This is the first genetically engineered animal that would be introduced to the food supply. So it's a big deal.
Atlantic salmon grows in fits and starts, but scientists found that adding genetic material from a Pacific salmon and an eel-like fish helps them grow round the clock.
This fish is indistinguishable from the traditional foods.
How can you say it's indistinguishable, though, and it clearly has a different genetic makeup than an Atlantic salmon?
But it tastes the same, it looks the same, and biology is exactly the same.
FDA scientists today agree.
Aqua Advantage salmon is Atlantic salmon, and food from Aqua Advantage salmon is as safe as food from other Atlantic salmon.
Still, critics claim more studies are needed on how the fish might affect humans and the environment. For instance, what happens if these altered fish somehow make it out into the wild? Would they breed with native salmon? Would they crowd out the wild fish? The company insists there are safeguards.
They're all sterile and they're raised in physically contained facilities.
But opponents say there's no guarantee all the fish will be sterile. And there's already one case study in how the best laid plans can go awry. Canola plants used to make canola oil were were genetically engineered to be resistant to weed killer. The problem, their seeds have now migrated from the company farms, carried by the wind, and perhaps by trucks taking them to market. They're now growing on the roadsides of North Dakota. Scientists worry they may create a hard-to-kill superweed. As for the genetically altered salmon, one question still on the table: will folks eat it? And will they even know what they're eating? Because the FDA has to decide if the fish will even carry a special label.
World Health Organization declaring a global health emergency.
We now expect the virus transmitted by mosquitoes, of course, could reach farther north, 30 states now.
Reading the latest now in the battle against Zika, the CDC says the virus has been linked to birth defects. Scientists hoping to use a new weapon, genetically modified mosquitoes. They're hot about this fear. They're dead set against using this neighborhood in an experiment. They're trying to jam this down our throat without our consent. And they're warning that your neighborhood could be next. Genetically modified mosquitoes to target insects that could spread the virus. But that plan is raising concerns in the neighborhood where they may be tested. And ABC's Steve Wilson is on the scene in Key West, Florida. Good morning, Steve. Good morning to you, George. They're hot about this fear. The families understand the effort to fight Zika and other mosquito-borne diseases, but they're dead set against using this neighborhood and their children in an experiment with genetically modified bugs.
There is news tonight about the quest for the perfect apple, one that doesn't brown when you slice it. Sounds too good to be true? Well, it's not, and it's coming to a grocery store near you. Here's ABC's Gloria Riviera.
Watch this, a time-lapse video of two seemingly identical apples. With time, the apple on the left browning as normal after cut, bruised, or bitten. The apple on the right still perfect. The difference, science. The apples are a brainchild of a small Canadian company, Okanagan Specialty Fruits. They say their Arctic apples, a version of the Granny Smith and Golden Delicious, have a suppressed enzyme that delays browning. It will still rot eventually, but stay cosmetically superior for longer. This week, the U.S. Department of Agriculture approved the Arctic apples, a genetically modified non-browning apple, deciding that they are not likely to have a significant impact on the human environment. We asked customers, how do you like them apples?
If I knew up front, I probably wouldn't have a problem with it. In fact, I'm a bit intrigued to try it.
I don't mind that they turn brown. I mean, you can do the same thing with lemon juice to keep them from browning.
Critics are concerned about misleading customers who may accidentally bite into an apple that is much older than it appears. Already, 80% of the corn and soybeans Americans consume are genetically altered. But as for these Arctic apples, the earliest you would see them in your grocery store would be 2017.
There's a lot there. But would you say that animals, fruit, fish, everything that lives is being corrupted? Is it pure? Man's going hell-bent to make that happen. And if you notice, it's being led by non-God-fearing countries. China, Australia. I mean, you can just go down the line where they're going crazy, and even this country's starting to go that way. So the point of it is, in the days of Noah, is it perfect? No. We are totally modifying and manipulating God's creation. What's going to be the impact? Jesus said. So, let's talk about creation of life. We saw an egg and a sperm create a living organism with a bright light. Well, now man has figured out a way to, guess what? Create life without the human
Sperm
sperm.
has been grown in a lab and then used to create offspring for the very first time, marking a major scientific breakthrough which could spell out the end to male infertility. Now, we're talking about Chinese scientists here. They're claiming that they've coaxed embryonic mice stem cells into sperm cells, which were implanted into eggs. The mice then produced a healthy offspring from the lab-created sperm. Now, this means a lot of things. First of all, male infertility,
affects 15% of all fertility cases. That's what we're talking about here. And sometimes men don't produce sperm, and the fact that they've created a sperm cell that can then reproduce and create a mice baby, a mouse baby, is really, I'm sorry, I'm getting really excited about the prospect of this because it affects a lot of people. And we're talking about fertility here. What do you make of this?
I think it's amazing. I think it's a huge breakthrough. And I always get so excited every time I get news of some organ or a sperm or whatever it is being grown in the lab. To me, that's the future. Like, that's the direction we're going in. So every time I hear an update on that, I think it's so interesting. It's going to carry your genes, presumably.
Right. I mean,
so
pretty much.
That's
so
awesome.
half of the DNA, the mitochondrial DNA, comes from the sperm, and the fact that they're able to take a stem cell, an embryonic stem cell, they have to mix it with the cocktail drugs, and of course a testicular cell. The study's in depth. I encourage you to take a look at it because it's really interesting. You know, prior to reading this, and it was actually insinuated in these articles, women don't realize how difficult it is to create sperm. In fact, I want to talk about that for a second. One of these researchers, this is what he had to say about sperm production. Each sperm takes 10 weeks to make and must go through a sequence of major changes. For example, halving the number of chromosomes. If it is to be totally fertile, to engineer this in a laboratory is a huge challenge. The fact that they're doing this, Nick, I mean, we're one step away from biogenetically engineering humans, but still good.
We also, though, if I can be a little cynical here, we have to understand that things oftentimes are easily done in the laboratory with mice. Seldom times do we have the opportunity to replicate that on humans, and it's a lot difficult, completely different, right? So, yes, this is a huge breakthrough because we got it down in the mouse, for example, but at the same time, we're a very long way from having the same results being seen in humans.
Well, this could affect men who suffer from mumps.
I'm just going to move on from that because it's getting to be close to 8:30, but you're getting the idea here. There's a lot of research. Starts out for the good, but what's going to happen when you get evil people involved in this stuff? There's no sanctity of what God has created. Now let's look at animal-human, the chromas, as we talked about a little bit. Bob mentioned a little bit about that, but basically this is where you grow human cells in animals for various reasons. So let's find out where that sits today.
This is it. Human stem cells being injected into a pig's embryo. For the first time, scientists are seeing how our cells grow and interact inside an animal. This pig, the first known chimera.
The ultimate goal for this type of research is to whether we can use this technology to generate human teeth and organs that can give us a lot of unique insight.
Right now, scientists are nowhere near that goal, but recent studies have shown how that process can work using gene editing. Scientists would delete a specific piece of an animal's DNA, so its embryo wouldn't be able to grow a heart, for example. Then they'd inject that embryo with human stem cells, which move into the void and grow a human heart. The embryo would then be put back inside the animal to develop.
The possibility of growing human cells and tissues inside animals is an exciting first step for the dream of generating tissues and organs for transplantation in humans. We're still far away.
To date, everything the researchers have done is legal, but what they're doing is highly controversial. The embryo is only being allowed to develop for 28 days. It's not allowed to be born. Bioethicists say this research raises a lot of uneasy questions.
The boundaries between what it means to be human and what it means to be non-human can become quite blurred. Also, it's not great for the animals. There's risk for them. There's no benefits for them. It may help a lot of people, but the moral status associated with this is a deep, deep, deep question.
That word, chimera, from Greek mythology is meant to elicit fear. But the scientists argue they are trying to solve a problem and save lives, not create a monster. Christine Dirac, CBC News, Toronto.
How about that, guys? Only 29 weeks? What happens if it goes beyond that? And who's going to prevent them?
Oops, we forgot that one.
Exactly. Now we're talking about GMO babies. So what I'm going to get into now is this CRISPR technique. This is something I talked about a couple years ago. It's the way in which people have found a way to manipulate the DNA. And now I'm going to show you a slide that's 4 minutes long that's going to talk about 10 events that happened in 2017 that are being used. And just think about, in the days of Noah, is it a pure line here? So let's...
Thank you for the visit. PsyQ is science made simple for the rest of us. Enjoy. What is CRISPR? CRISPR is the revolutionary new gene editing method that has the potential to completely transform our future and our society. The CRISPR technology was only developed about 8 decades ago, but research has shown tremendous progress in the last few years. Headlines about CRISPR breakthroughs have been more and more frequent recently. CRISPR-Cas9, short for clustered regularly interspaced short palindromic repeats, is a system for gene modification. With this new, revolutionary technology, you would be able to specifically change embedded sequences of your DNA. You could completely delete certain genes from a cell, or simply alter the amounts of it, or create a mutation inside a gene by changing the base pairs, tiny pieces of DNA. This process is called genetic engineering. And that brings us to the 10 biggest CRISPR breakthroughs of 2017. 1. CRISPR was used to remove HIV from living animals. Scientists in Pittsburgh were able to completely remove HIV from living mice using CRISPR. All of the test animals survived, and the tumors were shrunk, and also didn't spread any further. 2. The first human embryo was successfully genetically edited with CRISPR. Just last week, news broke that scientists had successfully performed genetic engineering on a human embryo. The researchers were modifying one-cell embryo DNA to remove a gene that is known to cause a specific heart disease. 3. CRISPR could slow down cancer growth. CRISPR was used to target the hybrid fusion, which is the command center of cancer, and is responsible for excessive tumor growth. Scientists were able to create a cancer-annihilating gene that shrunk tumors in mice who were carrying human prostate and liver cancer cells. 4. CRISPR could eventually create new organisms. Researchers have used E. coli bacteria to create semi-synthetic organisms. This was done by breeding them with an anomalous 6-letter genetic code instead of the regular 4-letter one. 5. CRISPR could reduce some of the world's most deadly diseases. Mosquitoes carry diseases like malaria, which are some of the most deadly illnesses on the planet. Scientists have been working on editing the fertility genes in mosquitoes, therefore limiting their spread. The success was also due to CRISPR's feature of changing multiple areas in genetic code at the same time. 6. CRISPR could offer a cure for Huntington's disease. Researchers have been using CRISPR to edit out Huntington's disease from mice and were able to reverse the symptomatic progression. This technique could also be applied to humans soon. 7. CRISPR can be used to modify viruses that kill antibiotic-resistant bacteria. The threat of antibiotic-resistant bacteria is growing, and CRISPR might finally provide a solution to this problem. Researchers were able to trigger self-destructive mechanisms in those bacteria by adding antibiotic-resistant gene sequences into bacteriophage viruses. 8. CRISPR could also revolutionize DNA storage. The idea of storing data in our DNA sounds quite crazy, but it's becoming a reality soon, and the concept is very interesting. Researchers were able to use the DNA of live bacteria to store a GIF using CRISPR, and then sequence the bacteria to successfully reconstitute the file. 9. CRISPR is biofuel. CRISPR is so versatile that there are even use cases out of the medical biology field. Scientists have discovered that genetic engineering in a specific yeast strain could lead to new precursors for sustainable biofuels. They were able to engineer certain algae that could double the biofuel material as their natural counterparts. 10. And lastly, there are also new CRISPR regulations to ensure safety. The DARPA, U.S. Defense Advanced Research Projects Agency, has taken upon the task of ensuring the accuracy and safety of CRISPR editing methods. To do that, they have invested $65 million in a new project called Safe Genes. The main objective of this initiative are to avoid misuse, which has always been a major concern regarding the CRISPR technology. And in conclusion, the CRISPR gene editing technique has only been around for a few years, yet we have seen some dramatic improvements. This list of 10 milestones are only about events that happened this year. The breakthroughs range from HIV and cancer research to gene editing in living organisms and prevention of deadly illnesses. If you enjoyed the video, please like, subscribe.
So what do you think of that, guys? Now man has the ability to create what he wants. And obviously, we saw all the good things that that could do. But imagine the Chinese guy that's trying to develop, a system to pick and choose intelligence. Or imagine anything else. And with wealth being established now in this world of being put around only a few people, families, shall we say, it becomes a very interesting corruption capability, because we know 100% power is 100% corruptibility. So we now have a strain of DNA that is being entirely corrupted. So now let's go into, there's a few, you guys want to stick around for a few more? I got 4 more.
Go for it.
So let's look at synthetic brain synapse. I talked about the brain series. So now the synapse is the integration where all the communication goes. So guess what man's come up with? A way to synthesize that. And that's going to be used to interface between man and machine. Cyborgs. Skynet. The Terminator. But I will not have any Terminator music going. Maybe I should.
Welcome to the Science Channel. My name is Ann Wilson. I'm a podcaster of this channel. Each person's brain has billions of neurons and trillions of synapses, a neural assembly that's shaped by time, environment, and experience in a way that is unique to everyone. Now, researchers have taken inspiration from the inner workings of this convoluted organ to develop an artificial synapse that they say is capable of learning autonomously. They have even modeled the device, which is considered the next step in the creation of more complex circuits. The study is published in Nature Communications. The team created a nanoscale device called a memristor, whose resistance depends on the electrical signals it has previously received. The idea of the memristor is not new. It was first conceptualized in the 1970s and subsequently built in 2008. This study, however, takes it to the next level. The idea of the memristor is to create an electronic equivalent of the brain's neurons and synapses, the biological wiring that is able to process and store information with incredible efficiency. Simply put, the synapse is the junction between 2 nerve cells that opens or shuts depending on the nerve impulses that reach it. Neurotransmitters cross that gap to pass the impulses on to the next neuron. Every time that crossing is made, the connection gets stronger and more efficient. To achieve a biomimetic version of this, an ultra-thin ferroelectric film was sandwiched between 2 electrodes, whose resistance can be tuned using voltage pulses. Thus, its plasticity, the ability to change and learn, is achieved via conductance. Low resistance corresponds to a strong synaptic connection, and high resistance to a weak connection. Content 1,491,493,470 170,403,140,249 to 1 to 900x60. The team then made a model of the device, and their simulations show that arrays of ferroelectric nanosynapses can autonomously learn to recognize patterns in a predictable way, opening the path towards unsupervised learning and spiking neural networks. Essentially, the work takes us closer towards improving the speed at which artificial neural networks learn and adapt. Artificial intelligence, AI, systems have developed a lot in the last few years, with Google's DeepMind and AlphaGo among the most popular examples. However, the brain is an incredibly intelligent machine and we are nowhere near replicating its sophistication. Even as you read this, neurons in your brain are firing a frenzy of electrical impulses and connecting to each other in ever-changing configurations. Such efficiency is a much sought-after goal in the creation of artificial brains. As the authors note, we are inching ever closer to an AI future. These results pave the way toward low-power hardware implementations of billions of reliable and predictable artificial synapses, such as deep neural networks, in future brain-inspired computers. Image and text. Artist's impression of the electronic synapse. The particles represent electrons, the flow of which depends on the ferroelectric domain structure. Credit. Sword Buoyancy and R.S. Bales Physics Joint Research Unit.
Did you get that? It's getting more knowledgeable, more creation, more the differential between, animals, humans, machines is becoming blurred. Now, as Bob mentioned, cloning. This just happened in the last few weeks. Well, they reported it. Obviously, this has been going on. So, this is a minute and a half. Cute monkeys. Notice the names, where it's being done. Christians are being killed. Churches are being pulled over. So, now we have cloning. We've gone from sheep to monkeys. Where do you think the next step is? As the rich and anyone with means wants to live forever. And what did God say about the Tower of Babel? They're trying to mimic us. Time to get rid of them. And that was an amazing time. So now I have one more event I'll talk about. 2 years ago I talked about the first head transplant that was about to happen. It's happened.
World's first head transplant is success after 19-hour operation. First ever head transplant. Written by Jimmy Russell. The groundbreaking operation took a team of surgeons 19 hours and has allowed the patient to be cancer-free. Johannesburg, South Africa, a 36-year-old man has undergone the world's first successful head transplant. The groundbreaking operation took a team of surgeons 19 hours to complete and has allowed the patient to be cancer-free. Paul Horner, who was diagnosed with bone cancer 5 years ago, was on the verge of death when he was approved for the controversial and possibly deadly operation. Dr. Tom Downey, who was part of the South African team who carried out the operation, told CNN he is thrilled about the results. It's a massive breakthrough, Downey said. We've proved that it can be done. We can give someone a brand new body that is just as good or better than their previous one. The success of this operation leads to infinite possibilities. Surgeons at Charlotte Max's Johannesburg Academic Hospital in Johannesburg carried out the operation in February, but waited until they could confirm it was successful before they made any public statement. Downey spoke to reporters about the complexity behind the first ever head transplant. This procedure is another excellent example of how medical research, technical know-how, and patient-centered care can be combined in the quest to relieve human suffering. The operation was led by Professor Myron Dennis and took place on February 10th of this year. Our goal is for Horner to be fully functional in 2 years, and so far we are very pleased by his rapid recovery, said Dennis. Before the operation, Horner's body was riddled with cancer and he had less than a month to live. We were fortunate enough to find a donor body, a 21-year-old man who has been brain dead from a serious car accident that happened in 2012. The boy's body worked just fine, but his brain was not functioning whatsoever, and there was absolutely no chance of recovery. Dennis continued. We received approval from the young man's parents to use their son's body to do the operation. They were extremely happy their son could save a life even in the vegetated state that he was in. Doctors say Horner has made an 85% recovery, walking, talking, and doing the normal things a healthy individual does. The Charlotte Max Johannesburg Academic Hospital, nicknamed Jobber Jensen accredited General Hospital in Vartum, Johannesburg, South Africa. It has 1,088 beds. The hospital's professional and support staff exceeds 4,000 people. Even though Horner lives in the United States, the first of its kind operation had to be done overseas in a location where the medical guidelines are not as strict. Related topic: how to receive HGH testosterone therapy. HGH is the miracle fountain of age wonder drug that everyone is talking about. But without the proper insurance, it is extremely expensive. www.hght.com makes it possible for you to get this amazing drug without a doctor's visit and at an extremely affordable price. Learn more about HGH and get feeling better today. The truth can change your life. Channel to raise the vibration.
Well, guys, that was a commercial, obviously. But the point of it is, there was another one scheduled in China. I don't know what happened to that one. But you notice it's being done outside. And this was a patient that was brain dead. Well, we all know where this can go. I mean, in China, there's documented cases where, human organs have been harvested from prisoners. So we know where this can go. But the point of it is, if God is up there watching this going on, how long is he going to allow it? As in the days of Noah, corrupt? So, man this is like I was saying in Genesis, has become like one of us. Knowing good and evil, now he can reach his hand and touch the tree and become immortal. God's not going to allow that to happen. It's already been discussed one time. Jesus said, as in the days of Noah. So to conclude, the Bible is his revelation to us. He told us what he's going to do. We need to be watchmen. And I would say we're becoming very much like in the days of Noah. The bloodline, everything's being corrupted. Our children are being corrupted. Our food's being corrupted. The DNA is being corrupted. It's a fallen world, which we know, but it's going beyond just you and I. It's now becoming created monsters, shall we say. So, that kind of concludes the update as of 2018 January. And you can see we've come a long way in just 18 months. And what's another 18 months going to produce? Right? So, it's scary, but it's also very exciting knowing that God is with us as we speak. Many of these things can work for the benefit. I have a kid that's got Parkinson's. There's a benefit to that, but at what cost? Right? And that's the issue from a medical standpoint. And if you're a military complex, what can you gain as an advantage if you do this? And right now, the CRISPR I mentioned, a lot of people in the U.S., a lot of the scientists are very irritated because they have all these restrictions. Where if they go overseas, China, South Africa, there are no restrictions. Those countries don't have a God constitution. So it's getting out of control. So I just think we need to pray, we need to, be aware, and we need to be able to share this kind of stuff because it is frightening. And if there was no God, where's this going to end up? Right? So with that said, let's end the evening, and I thank you all.